everybody. Welcome to this month's edition of the Innovate seminar, run jointly between the National Society of Black Physicists, NSBP, and the Kavli Institute for Theoretical Physics at UC Santa Barbara, KITP. I first want to thank both our speaker, Dr. Ilenna Jones, and our affiliates at KITP, mainly Lisa and Ilenna, and the rest of the NSBP team who's made this event happen. So without further ado, I think I'm going to introduce our speaker, Dr. Ilenna Jones. Dr. Jones is a research fellow at the Kempner Institute for the Study of Natural and Artificial Intelligence at Harvard University. Her research interests include neuronal biophysics and computation, model optimization, and neuroscience for AI. She received her BA in neuroscience in 2015 from Dartmouth College, and in 2023, she received her PhD in neuroscience from the University of Pennsylvania as a part of Conrad Corden's laboratory. Ilenna began her position as a research fellow in the Kempner Institute in 2023, and today she's going to be presenting on quantifying the impact of dendritic properties on neuronal computation. So without further ado, I'm going to hand it over to Dr. Jones.
Thank you, Carlton. Yes, thank you, everyone. It's very nice to be here and presenting to the NSBP. Today I'll be talking a bit about quantifying the impact of dendritic properties in neuronal computation, but first a little bit about myself. So I began in, as Carlton had explained, I did my BA in neuroscience, and there I focused on cellular molecular neuroscience, thinking about psychiatric neuroepigenetics, if that mouthful means anything. But there I also nurtured an interest in computational neuroscience that I finally delved deeply into when I joined Conrad Corden's lab at the University of Pennsylvania, where I did my PhD starting in 2017 and joined in 2018, where I was merging some of the fields and principles of machine learning with neuroscience. And then I defended my thesis in 2023, and I began as a Kempner Fellow in November 2023, and now I'm here. So without further ado, we know that brains enable intelligent behavior, and there are many aspects to the brain in terms of its organization, its connection to the rest of the body, and it's also known that in the human brain, at least, there are about 83 billion neurons in the brain. And would it not be interesting then to get a sense of, to know what the brain is capable of, or how that intelligent behavior is possible? Perhaps it would be interesting to look down at the individual neurons themselves and see what they're capable of. Since there are so many of them, what might that further then enable us to do when you start thinking about multineuronal or higher organization of these neuronal systems? So I study single neuron computation. What does this mean? Well, a single neuron, I define as a cell in the nervous system that consists of a soma, an axon, and a dendrite. And right here, I have two images of neurons, where this dark patch here is the soma. This line here and here is the axon, and the soma is what collects information from this intricate tree structure, the dendritic tree, which receives inputs via synapses from other neurons to this neuron, collects that information, electrical information, at the soma, and when that information or when that electrical activity reaches a threshold, it triggers an action potential, which is a spike that travels down the axon to another neuron where it might transmit information. So what do I mean by computation? I defined computation as information processing by a physical system for the purpose of accomplishing a task. So for example, if you were to have a physical system represented by some function f, an input would go into this physical system and map that input to an output. And that is what I mean by information processing. So going back, we were wondering what a single neuron is computationally capable of, and this definition of computation. So what do I mean by computational capability, and how might I measure it, and how might I measure it for a single neuron? So how would we measure computational capability? So going to the field of machine learning and deep learning, there's a framework called supervised learning, which consists of 3 components that might help us do this. And those 3 components are a model, a specific task, and a learning rule. And for example, here I have an artificial neural network taken from the field of deep learning, and it can be, for all intents and purposes, our system defined by F, or a model defined by F. It receives an input X and produces the output Y parameterized by a set of parameters theta, which would be represented by each of these black lines connecting all of the nodes or neurons in the neural network. So what is this model doing? This model could be doing a task. For example, if I gave it a pixel input of the handwritten digit number 3, it's 28 by 28 black and white pixel image, gave it to a neural network, it might then compute an output. Perhaps when it came to this task, it would be the classification of the input to its correct label, which is 3. So how would this model then be able to perform this task if we don't know what the parameters theta are? Well, you can do so by using a learning rule. So for example, if I, for all intents and purposes, had a model that only had 2 parameters, or a neuron or node with only 2 parameters, defined here by this plane, anywhere along this plane could correspond to a particular model. And that model can then be, the performance of that model on a task can then be quantified using an objective function, for which the quantity for that return would give us this third axis. And it will perform, the learning rule can then take the information from the objective function and make certain decisions on how the parameters of the model should be changed in order to ascend to the best performance of the objective function for a particular model in mind. And that optimal performance, I will go ahead and call the computational capability, or the upper bound of task performance of a model. So this is what I mean by computational capability. So then going back to framing the question, can I measure this computational capability of single neurons using this supervised learning framework? But of course, going back to this, we have this whole framework, but what do I mean by a single neuron? And what neuron model would I be using? So we can go back and ask, what is a good model of a neuron? We can actually get a little bit more fundamental. What is a good model? Norbert Wiener says the best material model of a cat is another, or preferably the same cat. However, we know that abstraction is necessary to make useful models. So you have to compromise. So for example, I'm in Boston, and say I wanted to navigate to my workplace for the first time from home. I could ask you, can you give me a map of Boston? And if you gave me a map of Boston that was exactly a 1-to-1 representation of the entirety of Boston, that map would not be very useful for me because I could not hold it and look at it. At least it would represent Boston, but it would have too much detail. It needs abstraction, perhaps abstraction to reduce it to the size of something I can hold so that it can be useful. Another type of abstraction you might think of is how you might communicate or represent something. So for example, let's think of the cats again. cats are very, they have a lot of different kinds of behaviors, properties. They're very different, and they could even enjoy science. But we may not necessarily need to represent all of these details of a cat. Perhaps what you might need to do is abstract it to produce maybe something like a cartoon that represents only the necessary features to be able to communicate or represent what a cat is. So looking at this, abstraction is important, but how much abstraction is needed to make a useful model? Well, it depends on your question. So let's think of a question to think about this process. So when I'm in my apartment, I often need to vacuum to clean the place up. And when I turn on the vacuum and start vacuuming, it makes a loud noise, and my cats kind of just jump away out of the way and run away, startled. And for an easy question, let's ask, why is my cat moving away from the vacuum cleaner? And let's come up with a model of a cat that is most useful to explain this phenomenon. So we can think of the idea of what kind of models might be useful for this by thinking of it existing on a spectrum of abstraction, where on one end, you might want to use a full real cat, but that can be very taxing, costly. You have to feed it, you have to clean up its litter box, it needs lots of attention, and you may want to have something a little more easy to think about. So perhaps we want to go to maybe a cartoon representation of a cat with various properties that correspond to the target system. But why stop there? We could have the same kind of representation, but without ears or without a body. Or why don't we go even further? I could just draw this picture of a cat, call it a cat, and call it a day. Why would we need a model that has certain properties when we could go as cheap as possible and as abstract as possible? Of course, when you go all the way to a model that is that abstract, you might lose certain mechanisms or properties that might be useful for explanation. For example, my vacuum cleaner makes a sound, and a cat without ears would not be able to respond to any sound because it could not detect it. Or the cat runs away, but a cat without a body would not be able to move. It's just a disembodied head. So we do need certain properties to be able to make a good explanation from the model that we choose. So we can't just abstract everything. We need to include certain kinds of properties to be able to generate certain kinds of knowledge to answer certain kinds of questions. You can even get more detailed models of cats, depending on the question that you have in mind or what degree of detail you would need for satisfying the question. But of course, all models are wrong, but some are useful. So what degree of abstraction makes a useful neuron model, especially if our question is about computational capability? So in big, bold letters, I will have humility. I don't know what properties of a neuron are going to be relevant to computational capability a priori. So let's try to make some best guesses and see if we can start somewhere. So there are many different kinds of neuron models. There are ion channel conductance models, GLM models, integrated fire models, and a lot of these are in the form of point neurons, where you can even go as abstract as the McCulloch-Pitts neuron, which is going to look very familiar if you're familiar with deep learning, which is simply a linear weighted sum of its inputs, summed up, and then put through some sort of nonlinearity to produce an output. And this neuron is actually the basis for deep networks in artificial intelligence systems. So you can also get a bit more detailed and represent certain kinds of properties of neurons, such as the electrical cable properties of a neuron with a cable model, or go even further and make even more biophysically detailed models, including ion channels, including cable dynamics, including many other things. And you can also put this on a spectrum of abstraction. On one end, the artificial neural network neuron, And on the other end, you can make very biophysically detailed models, and you can even push those detailed models to be even more detailed if you wanted to. But many scientists choose something a bit in the middle, where they take some properties but leave others. Now, one particular property that I want to focus on that I think is going to be useful for us to think about is the difference between these two types of models. On one side, you have point-neuron models, and on the other side, you have detailed models that include dendrites. This highlights the point-neuron assumption, where neuron models are without dendrites are making that assumption. And that's a very useful assumption. Many different models of neuroscience and multineuronal models in neuroscience are very useful making this assumption and have generated a lot of knowledge. However, we mustn't ignore the fact that dendrites are incredibly detailed and complex, and that making the point neuron assumption is removing these structures of neurons. And not only that, but there are many different properties of them as well. For example, not only are they intricate in their morphology, they also have various different kinds of conductances. So whether that be from other neurons through their synapses, or voltage-gated ion channels that may be intrinsically part of the dendritic tree that are gated by changes in from voltage depolarizations. There are even not only action potentials that come from neurons, like in many single point neuron model formulations, but there are also dendritic spikes, which would be left out from models without dendrites. For example, NMDA spikes, calcium spikes, and backpropagating action potentials that can occur when an action potential is released from the axon initial segment. And not only is their signal moving down toward the next neuron, there's actually current flowing back up through the dendritic tree backward. There's also synapse location and clustering, and there are also computations, like the possibility of producing and XOR functions in the dendritic tree. And lastly, all of these things can change with experience and over time. So let's just say that maybe dendrites are relevant. So what properties might I choose then for my neuron model? If we're thinking about information processing, and dendrites seem to be part of information processing, perhaps we can get somewhere with this. So I didn't know what properties are relevant a priori, and dendritic properties are relevant to information processing. So if a relevant property seems to be more impactful for a neuron's computational capability, then which dendritic properties seem to be impacting this capability? So lastly, before I finally show you what neuron model I choose, I want to point out that certain kinds of mathematical formulation, all of these models are mathematical formulations. They're very abstracted in the fact that they are just relationships represented by math. But what makes them neuron models is that certain kinds of relationships and certain kinds of properties and states correspond to or are analogies of actual neuronal properties. And so what helps is that perhaps I can look at another mathematical formulation and argue for a strong enough analogy that allows me to then use that formulation as a new neuron model. So I suggest artificial neural networks with neuronal empirical details are neuron-like. So say I take an artificial neural network, this is a 2-layer one, and I want to add dendritic constraints, perhaps constraints as in constraints toward biological realism in some way. And as I add those constraints, I'll be stepping more and more toward biological realism in this artificial neural network that might then allow me to quantify the computational capability of the impacts of these properties on the computational capability of such a model. So using a supervised learning framework, I'll use these models, neural models, to then quantify how these models impact the computational capability of how these dendritic properties impact the computational capability of these single neuron models. All right, finally, results. So I have here 5 different kinds of constraints, and I'll talk about first the structural constraints and then the non-structural constraints. So first, I'm going to introduce structural constraints. So let's think about what a neuron's structured as. So here's a cartoon of a neuron with its dendritic tree. This is the cell body, the soma. This is the axon. And here are the dendritic subtrees. And I want to point you to an aspect of this, which is that dendritic trees are binary branching structures. At any point where there's branching, there are 2 child branches coming from one parent branch. Can we then, that is one property that I want to try testing. And then secondly, from the literature, there's evidence showing that on average, a presynaptic neuron, a neuron that's sending in signal to your target neuron, your postsynaptic neuron, the presynaptic neuron can synapse or make connections on average 4 times to your target neuron. And of course, that is not normally distributed around 4. It could be that there's many ones and a few 100, but let's just go with the number 4. And that synaptic repetition is also a structural constraint I want to try. So I take an artificial neural network and constrain it with binary branching structure so that it is more along like the shape of an actual neuron in its binary branching structure. And here, I'll break it down. The blue nodes correspond to the inputs. The white nodes correspond to the branches. So here you have 2 inputs, terminal inputs, coming onto a branch. And the black lines correspond to the connections between branches and inputs, whether that be synaptic weights for these black lines or axial conductances connecting 2 branches. And then pink is the soma, and that is where the output is. And secondly, for synaptic repetition, because we're not quite clear on how these synapses are repeated, and if that is in any way a uniform or universal pattern, we assume that the inputs are repeated on different separate subtrees, where the same identical input on one subtree will occur in the same exact place on all other subtrees. So for example, if there were 2 subtrees, the neuron 1 will synapse here and here, where 1 corresponds to that neuron 1's connection. Of course, you're probably, well, I'll move on. So the impact, we're going to test the impact of these properties. So we will have certain kinds of controls to compare these 2 experimental models to, and those controls will be a linear point neuron model, one that is very similar to the just a single artificial neural network neuron with no dendrites. It's just synapses onto a soma, and then there's an output. And then there's a fully connected neural network, which is just 2 layers deep. And then we have the 1 tree, right, where K is equal to the number of subtrees there are, or repetitions there are. And this only has the binary branching property. And then we have the K tree. And in this case, K is equal to 4. And it is a combination of the binary branching structure and the synaptic repetition. And if we do not want to have the binary branching structure, but we want to have the same number of parameters as the synaptic repetition constraint, we'll actually take the second layer of this fully connected neural network and increase that number so that the number of parameters, these black lines, are roughly equal to the number of black lines or parameters in this model. And those are models and controls. We'll be setting it to task for this binary branching structure. This binary classification task, where it's classifying between the images of a number 3 and the images of a number 5. And it is producing a binary input output 1 or 0. And finally, the results, where we have on the x-axis what kind of architecture we're using, k. So k equals 1 corresponding to this condition, and k being greater than 1 corresponding to this condition. And then finally LDA, which is a proxy for the linear point neuron model. And the y-axis is the accuracy or the performance on this task, where random chance would be 50%. So the linear point neuron model proxy performs pretty well, around 87% with these error bars for about n equals 10 different trials. And here in gray, you have the fully connected neural network or the two-layer neural network performing pretty well, around 95%. And here we have the K-tree or the one-tree model with just the binary branching constraint that has reduced performance. Now, when we add the synaptic repetition properties, we actually see an improvement not only from the gain in number of parameters, like in this gray line here of the fully connected network, but also we see a increase almost meeting the performance of the fully connected network here at a very unrealistic but still present architecture of k equals 32. So binary branching structure seems to reduce performance, but synaptic repetition improves performance. In conclusion, as a conclusion slide, it's not the conclusion slide, but just to talk about the structural constraints, our neuron-like artificial neural network models can consistently perform above the point neuron level. And this work does not necessarily support the use of the point neuron assumption in investigations of computational capability, because when you include dendrites, it seems like computational capability is impacted. Dendritic branching seems to reduce the performance of fully connected networks. And this could be due to the introduction of very high sparsity. Strict binary branching structures are very sparse in their connections. And synaptic repetition seems to improve the performance, and this may be because of an increased redundancy within the dendritic branches, which could allow for easier learning and more degrees of freedom to work with. So let's consider moving closer to biological realism by thinking about the non-structural constraints. So there are 3 others that I'll talk about, the first of which is the dendritic nonlinearity constraint. Now, I'll have you focus on the single node of a K-tree model. So one thing that I did not necessarily talk about is that each of these nodes have what is an activation, an activation function, where in deep learning, the activation functions tend to be some form of ReLU or leaky ReLU function, where a ReLU or leaky ReLU function means that if the input is negative, it will return an output that is 0 or close to 0. But if the input is positive, it will return the identity or the same output as the input. And this is not very biologically realistic. Negative current or negative voltage does not necessarily zero out immediately once it reaches a new branch. So we tried something different and introduced a dendritic nonlinearity. So it's formulated as such where you don't have to look too closely at the math, but you can see that we just included a linear function, which comes from the conductances, axial conductances multiplied by the voltage of the adjacent compartments, a sodium, calcium, and potassium term that come from the input voltage curves of these certain kinds of channels. And it's just simply adding all of these up to get the dendritic nonlinearity function. I call it the NACAK. I think it could be named something better. But what we really see is kind of a line with a bump in it. And it's not technically not nonlinear, but it looks pretty linear to me. But if you remove the massive linear component, you can actually see that the nonlinearity is actually very rich here as it changes from inputs from a negative and very positive. So what do I want to do with this? I want to then take this dendritic nonlinearity and put it and repeat the previous experiment with the structural properties and compare it to other activation functions used in deep learning. And that might be the ReLU, which is in red, leaky ReLU in pink, sigmoid function in purple, and swish in green, where swish is kind of like a ReLU function, but smooth. So what if we replace the ReLU activation function with the dendritic nonlinearity? So what I'll do is I take the binary tree model with the synaptic repetitions and repeat the same experiment, but with different activation functions. So this is a bit noisy, but I'll walk you through it. So red and pink are ReLU and leaky ReLU, and you can see that they perform down here below the dendritic nonlinearity, the NACAK function, which is up here with this binary branching tree. And the swish function actually does pretty well as well. Why might that be? Well, let's try to figure that out after we compare it to the control. So what if we did the same experiment, but with the fully connected control? Again, it's going to be parameter size matched by changing the hidden layer width. And you can see here, red and pink, you see ReLU actually performs much better than this dendritic nonlinearity in gray and even swish. Why might this be? Well, we can think about sparsity again. So if you think about it, ReLU and leaky ReLU introduce sparsity to some of the computations that are happening in the network. And adding a little bit of sparsity is very beneficial, but having too much sparsity, where you have these nonlinearities, ReLU and leaky ReLU, with an already sparse binary branching structure, you get something that performs not as well than as a bit of sparsity to a very densely connected network. And also, if you look at the y-axes, you can see in gray here and here, they're actually the same. So what we're actually seeing is that ReLU is performing worse in this binary branching structure, but better in the densely connected one, whereas the dendritic nonlinearity is performing just about the same. And so it may be that having a smoother function like the dendritic nonlinearity and swish even helps this binary branching tree structure perform well. So, and then now for the non-structural constraints, synaptic nonlinearity and the non-negative weights. So just to talk through them without going into too much depth, in our previous architectures, there were no particular synaptic nonlinearities present. So if you put in an input, which is going to be between 0 and 1, it would go through the network as some value between 0 and 1. And if you were to introduce a synaptic nonlinearity, there's certain kinds of aspects to this nonlinearity that we defined, but what should be taken from this is that it maps the 0 to 1 input to something between -70 and 50. And it's not a direct mapping, but that is, I think, the most important thing to take away. from the synaptic nonlinearity in this case. And then for the next constraint, you have these non-negative weights. So if you remember before, I said that these black lines between these dendritic branch nodes could correspond to axial conductance. Well, conductance is non-negative, but in artificial neural networks, they're real value. They can be negative. So we constrain them to be non-negative here. So that is to step more toward biological realism. So we add these both, kind of skipping to the end, we add both of them, and we have all of these different conditions, but I'm only going to have you focus on two, where on one side is the experimental condition, where we're taking a binary branching structure that we were talking about before, and the control condition, which is the fully connected neural network structure, the densely connected one with two layers. And these are the two conditions that I want you to focus on. Otherwise, they have all the rest of the other shared properties. All of the dendritic constraints are going to be added to each of these structures. And we're going to see how they perform in the same setup. So it's going to be a bit of a noisy figure, but just look for red and orange. So you can see that the control condition performs here, and then the experimental condition with the binary branching structure actually performs a bit better. Why might this be? Well, there are many different things we added. And one thing that we were thinking about was how some of these properties seem to interact to contribute to this increase in performance. And that is the dendritic nonlinearity interacting with the synaptic nonlinearity. So if you remember the synaptic nonlinearity, the input is mapped to be somewhere between -70 and 50, which is in millivolts for an actual neuron. In the dendritic nonlinearity, if you look closer at the x-axis, you can see that it actually is very wide, between -70 and 50 as well, is what would be informative, or would take as much use of the full size of the dendritic nonlinearity. Whereas if the input was between 0 and 1, it would only be in the small range of this small curve space here, or the small curve with the linear component here. So it takes advantage of more of the nonlinearity. So from here, I can say that this model, which uses all of the biological constraints, performs better than the model that has all of the biological constraints except the binary branching structure, something that actually flips our initial result. So to conclude, there's several limitations with this work. There were a lot of abstractions. So for example, we use completely symmetric dendritic trees. Dendritic trees are very asymmetrical. They have all sorts of funky structures. And some work by Oja 2022 thinks about the asymmetric dendritic trees and how that might contribute to computational performance. Our model is a feed-forward artificial neural network, and they're static. There's just one step to the output, and neurons are dynamic, where it would take many steps to find some steady state and so on. So further work of this kind must be done with temporally dynamic dendritic models. And lastly, but not the only, this is not a comprehensive list of limitations. A computer vision task is not likely what single neurons do. It is not likely that a single neuron is receiving only the pixel inputs of a 28 by 28 black and white image of the number 3. Further work should be done to identify then complex tasks that single neurons should do in the brain. So in conclusion, we developed a method for determining the impact and relevance of qualitative properties for a computational capability. We demonstrated that dendritic properties synergize to have more impact on computational capability. And this work suggests that the point neuron assumption, where you remove dendrites may be biasing the study of intelligence at the neuronal and multineuronal levels. And biological realism should be included in explorations of computational capability in single neurons and beyond. With that, thank you so much to the NSBP for hosting me. Thank you to Penn, where I did my PhD, the Cording Lab, and all of the people in it. And if you have any questions, I can be found on Twitter, but also my email here at Harvard is also a great way to contact me. Thank you very much.
Thank you so much. This was a really great talk. And I guess before we go further, I do want to say if you have any questions, you can use the Q&A feature at the bottom of the panel. We already have one question in the Q&A, so I'm going to read this and I'm going to ask my own question. Alan Pierre-Louis asks, can you use a weighted Markov chain model to represent neurons in their respective firing potentials? And he says, as I understand, that is typically mediated by membrane properties, but perhaps Markov methods could help model that in a helpful manner.
Yes, so to answer that question, should I click answer live? I don't know.
Oh, I can do it.
Okay. So to answer that question, I do think that weighted Markov chain models could be a way to model neurons, especially if you're trying to get at some of the dynamic or temporal aspects. My understanding of Markov chains models is that there's some sort of discreteness. Is that right, Carlton?
I'm not entirely sure myself, but
That's all
I
right.
think, yeah.
So my understanding of Markov chains is that you're going from one discrete state to the next. And I do understand that neurons, or well, rather a lot of things in the real world are going to have some sort of continuous dynamics or continuous temporal dynamics. So using discrete dynamics could be potentially a nice abstraction. Yes, thank you. You did say they're discrete. So yes, I'm wondering if that could be useful. I think that could be a good next step for trying to get at some of these temporal aspects of these neurons. But I'm not sure how else to answer that further. I hope that helps.
Thank you. Appreciate it.
Mm-hmm.
So I guess my own question was, I guess, obviously, like you, I guess I was wondering, how should we interpret the weights in this model? Is it like the weights are like the connections between like the different branches and then the branches to the soma, correct?
Yes, so apologies for skipping through this, but here's a lovely diagram here. So the black lines, which correspond to the weights, can be interpreted two ways. One way is if it is a weight connecting an input to a white circle node branch, that would be considered, in this analogy, a synaptic weight.
Mm.
And if this line corresponds to two wide circles or two dendritic branch nodes, then that would be considered an axial conductance weight. And axial conductances could correspond to, or could be corresponding to the diameters of the cables that each branch has. We don't really put any constraints, or we're very agnostic about what weight, the smoothness of these weights, for example. You might have a very large conductance, which corresponds to a certain diameter here, and then a very small conductance or a very different diameter there. And we don't have any smoothness constraints. which could be a very interesting way to try to make this a bit more biologically realistic. Because it kind of would look like a bit of a Franken tree, where you have like little spindly branches and then big thick branches, and it could be something to explore further.
I guess maybe this is a bit of a follow-up question. But in your objective, well, maybe not your objective function, but I remember it was you added all up the contributions of each of the different kinds of ion channels. So like the potassium, the calcium, and the sodium. And this was to define the nonlinearity. I'm wondering, I guess this is what's your educated guess on how variable like the contributions from the different like ion channels are like in biological systems? Is there like reason to believe that they're like highly like stereotyped, or there is like a lot of variability in like the contributions?
Yeah, that's a fantastic question. My understanding is that it is a bit difficult to be able to quantify this. I think you would need some certain kinds of imaging techniques in accordance with labeling techniques to be able to look at particular channels and where they ended up in the dendritic tree, and then further quantifying that. But even then, doing this for many neurons or doing it consistently is a bit of a challenge. So when it comes to this model, I set DNA. So I set the conductance terms here, which really just is a linear weighted sum of these curves, and I set them all equal to 1. However, in the code base, which is something I've been meaning to get to, I've made this potentially learnable. So each of these terms learnable. So it would be very interesting to go back and see which nodes or which clusters of nodes perhaps had more potassium or more sodium or less calcium. Could there be hotspots? Could there be a trend toward more potassium conductances as you got closer or further from the soma? All of these are valid questions and could be quite interesting to follow up on. So great question.
Thank you. And I want to remind people that if you have any questions, you can use the Q&A feature. But I think my last question is probably the last point in your conclusion slide, which is what do you think is fair to say the computation that a single neuron can do? Or
Yes.
like the type of task you think like single neuron would like do if it used like a dendritic features?
Yes. So there's a bit of work on this. There's, I think, going all the way back to Wilfred Rall's work, I think it's Wilfred Rall's work, where you can have direction selectivity in the retina that can be picked up by dendrites in the retina kind of spreading out, fanning out. And if you have motion coming in toward the soma and going in a proximal direction, you can get a buildup of activity. But if you go away from the soma in a particular other direction, you can get almost insignificant activity. And that's a phenomenon that's well documented. And I feel like that's one type of kind of to be honest, I'm not quite sure how to best define the difference between phenomena and computation. I think that comes down to a little bit of philosophy that I myself am trying to learn about. When it comes to other kinds of things, like what kinds of perhaps non-trivial tasks that single neurons could do, given that they have their dendrites, perhaps you might want to look at some aspects of the visual system. For example, if you have I'm kind of spitballing here a little bit, but I was thinking of like early parts of the visual system, perhaps even the LGN or before the V1. You might find that if you were able to find a good mapping of what is actually being received by the retina from the outside world, and then map that to, well, how does the LGN or even the V1 respond to that? And is there any kind of early processing that might lead to faster responses? For example, a rabbit darting under a bush because it saw a moving shadow, as opposed to a rabbit looking up in the sky and thinking, is that a bird? Is that a plane? It's a hawk, and it's going to get me. It's perhaps some sort of earlier timing or faster timing might come from computations earlier on in the visual system. But that's, of course, spitballing. I am curious to see what other people have thought about this.
Thank you. And this is, I think, our final question, and then we can wrap up since we're approaching the top of the hour. And this is from Chris Lynn, where Chris asks, you mentioned that neurons link with k equals 4 synapses on average, and you also mentioned that some neurons link with many more synapses. Do you have a guess for how this variation in k will impact computation?
Yeah, so it's synapse location is, I think, a very interesting question, and synaptic repetition is part of this question. Sometimes repetitions can be of synapses. In this case, I assume that the synapses are being replicated in distinct dendritic branches, completely separate from each other, before, if they were to have any kind of further downstream interaction, it would be at the soma. It could be that synapses could be on the same dendritic subtree, some degrees apart. And some synapses that are repeated can happen right next to each other, leading to a kind of, what do you say, leaning on certain kinds of nonlinearities that can exist in the dendritic tree that are voltage gated there. That can lead to superlinear summation of current inputs. So the location of these synapses is, I think, just as important as how many of these synapses there are, given that how many there are might also impact where are they in relation to each other. So I did mention a bit about the superlinearity. I mentioned before a bit about dendritic spikes. So you can actually, if you have enough of a depolarization of the dendritic membrane, you can trigger what are very powerful NMDA spikes, where certain kinds of channels only open when there's large enough voltage depolarization and they're receiving input from another neuron, leading to an influx of many cations and sodium and calcium that would be more likely to reach the soma than any single synaptic event. So having synapses or many more synapses all correlated in their activity, because they're all coming from the same neuron, might lead to a more salient signal or a salient impact of that particular neuron on the target neuron's activity, whether it might correlate with the presynaptic neuron or otherwise. So that's my best shot at answering that. Hope that was good enough, and that I'm leaving you with just as many answers as questions.
Awesome. Uh, so at this point, I really want to thank you, Ilenna, for giving a really great talk. It was phenomenal. I learned so much about dendrites in computation than I did before. And yeah, no, just like the diversity of like the single neuron models. Uh, and I want to thank our affiliates at KITP, Lisa and Ilenna, for setting this all up. I want to thank uh, uh, our NSB members of NSBP, uh, Alan and Stephen, and the rest of the student council for helping set this up as well. And yeah, do stay tuned for other iterations of the Innovate seminar. Yeah, and thanks to our audience for participating. Yeah.
Thank you. Great questions and great moderation. Thank you so much for inviting me.